Hello, this is Tros Viktorovich Pluskin. This is a review talk on conditioning and spawning strategies for eastern oysters, Crassostrea virginica. This work, I'm currently based out of University of Florida, though this work was done during my time at the Noank Aquaculture Cooperative Shellfish Hatchery. So to properly contextualize just how important refining oyster spawning and conditioning techniques in the hatchery is, you really have to have a brief understanding of the American East Coast oyster industry as a whole. This is an industry that reached a multi-million dollar scale between the colonial period up until its peak, about midway through the 19th century. And basically, what really allowed this industry to blossom was mass production and mass harvesting of oysters from wild populations of oyster reefs. So these oyster reefs would be dredged, oysters would be harvested, steamed, canned, and then their meat would be distributed through the rail lines throughout the entire continent of America. So oysters became a very cheap, very reliable, nutritious source of seafood that could be stored and distributed throughout the country. So what fueled this massive production really was kind of a hybrid approach to fisheries behavior. So the oysterman, the oyster fisher, was not only a harvester of the crop, but he also directly participated in the recruitment of the wild oyster reefs. It became the practice of the traditional oyster fishermen to collect all of the old shells from the shucking houses and from, after the shell had been consumed, and these shells were dried and collected and dumped onto specific sites during specific times of the year when these oystermen knew that there were wild populations of oysters that were spawning and a few weeks had passed and that there were then oyster larvae in the plankton that were ready to settle. So by refining these techniques from the colonial period, using knowledge of the Native Americans, and refining them onto the industrial scale by the time we had the 19th century, oystermen were really able to reliably re-recruit oysters in specific areas by moving shell around and by facilitating available ecological niches for wild plankton to settle. And this system was extremely productive and is still productive to this day. However, there were some issues that arose mainly in the 20th century, which completely destroyed this system, and thus the multi-million dollar mass production oyster system no longer exists on the east coast of the United States. And over the 20th century, these factors that really collapsed the wild oyster reefs were multifaceted. So we had hurricanes, which physically destroyed the reefs and scattered their broodstock. We had pollution arising mainly from the industrial sectors that had arisen across the river systems on the East Coast that were directly poisoning and killing the food, larvae, and broodstock of the oysters. We had the First and Second World Wars, which recruited heavy amounts of fishermen, including oystermen, and a lot of their old knowledge was lost on the battlefields of those wars. And then we also had the introduction of novel diseases from foreign waters, such as MSX and Dermo, which had their own chances to wipe out naive native oyster populations. So through all these mechanisms, we had a reduction of the wild oyster population, increased prevalence of novel diseases to wipe out their progeny, and then a reduction of the oystermen, which had facilitated their expansion and re-recruitment even when heavily harvested. So we had a huge collapse in the oyster industry, and where it once has been largely lost. And what has arisen in its place is this new 21st century American oyster marketing scheme, which is very in a lot of contrast to the old system. As opposed to mass production of oysters to be processed, canned in high concentration, and then sold for a relatively low price per oyster, we're going in the opposite direction. Instead of larger oysters, we're harvesting them much smaller. Instead of cooking them, we're serving them raw. Instead of selling them for a wholesale price, we're getting a premium price even at the wholesale level, so upwards of 70 cents a piece for an oyster. So we see here a complete transformation both in the product that is desired from the American oyster industry all the way to the consumer. The average oyster consumer does not crack open a can of oysters in America and consume it. They go to a restaurant and they receive the product raw and fresh in its own form. And now there is, as opposed to being a homogenized commodity, the oyster is now a highly specialized product where even the individual farm that it comes from gives it its own sort of brand power. So we see here a complete transformation towards the kind of product that we want in the 21st century raw bar market. And it is here where refining hatchery production is all the more important. And then here we briefly see one of the lasting hallmarks of this old industrial oyster era, where you have these advertisements that were put up everywhere and advertise the various major producers. But that old industry is very much gone in the history books. And what remains is something which is more or less much more reliant on hatchery raised production, not only for the need to produce disease resistant individuals that will be able to survive through grow out, but also the need to be able to actually produce oysters for the single shell raw bar market. And this slide summarizes where we've been and where we currently are and where we're going as far as the current standing of oyster aquaculture on the East Coast of the United States, where there are still players that depend on natural re-recruitment of oyster set using shell, but that is largely dwarfed towards the raw bar market, which depends on hatchery raised seed. And why are we so dependent on hatcheries nowadays? You know, if it's so much more expensive to operate a hatchery compared to wild production, and it's because of these reasons that we've seen, which is one, it supplements production in case of huge calamity in the wild populations. Any oyster farming family which has survived the old days has extreme fears of the next novel oyster disease, which will wipe out the current stocks that have been established to this day. So there are ongoing efforts on the academic and industrial level to produce more and more diverse strains of oysters with more and more increased competency towards a wider range of environmental conditions and pathogens. Most importantly is the fact that because of this reality of the new raw bar renaissance market, you can no longer afford to have the majority of your crop being produced through the natural way. In the wild, you have thousands, millions, trillions of pettivellager oyster larvae, many of them settling on the same piece of substrate all at once on each other. So you'll have these oyster clumps, which have 2, 3, 4, 5, 6, 7, 8 oysters all growing on top of each other, sharing the same substrate, if not growing on top of each other's shells. And even when broken up into individual pieces, these oysters are largely undesirable for the raw bar market because they have a regular shape. They do not look the same on the plate when it comes to the raw bar platter. People want a uniform shell shape and size. People usually want a deep cup, and people usually want a good deal of tumbling to go down with their oysters, because the less cuticle that you have, the thicker that shell is, the more it's been reinforced by the actual process of handling, the more easy it is to shuck. No one wants to have shards of thin shell being put into their $2.50 oyster. So it is imperative that oysters can be produced in the hatchery so that you can ensure that a single larvae is going to set on a single piece of substrate, so that no matter how much that oyster grows, no matter how big it gets, it's still going to be that one uniform size, which the market is desiring. When you have raw bar shuckers going through thousands of these things a day, they want them to have a relative uniform shape and ease of shucking and no destroyed, broken up shell inside the meat. And then, of course, also hatcheries will also facilitate in selective breeding, which we've gone over. And lastly, in this world where we do have mass expansions and spread of oyster genetics, hatcheries, especially local hatcheries, are paramount to being able to retain and keep track of localized genetics in case that there is a new novel calamity and we need to re-recruit genes from various populations to make new efficacious strains again. Having local hatcheries making local oysters is the only way to have an organized source of those genetics. And here's a quote from Victor Lusinoff, considered the father of American shellfish aquaculture when it comes to hatchery culture, at least. Another and extremely strong argument in favor of shellfish hatcheries is that by using them, we may, in some instances, do better than nature itself. In other words, instead of depending on nature to supply us with seed of a quality that we cannot control, hatcheries may provide the industries with oyster or clam seed of known, desirable characteristics. For example, by selecting certain individuals for breeding purposes, it may be possible to develop disease-resistant races of shellfish. Imagine what it would mean to the oyster industries of New Jersey, Delaware, and Virginia today if 15 or 25 years ago we had been able to breed oysters that were immune to the diseases of which, at present, is causing the industries of those states losses of millions of bushels of oysters. So we can see here back in the 60s that Lusinoff can see the future of what his and everyone else's work will do as far as opening the gates. By being able to produce oysters in the hatchery, we have a mechanism for being able to guide their genetics towards being able to overcome future perils, be them natural or be them induced by our own behavior. So we have here a tremendous tool that should be harnessed and continuously refined so that we do not have the destruction of the hardworking, all those hardworking people that lost their livelihoods with the old industry. Even though Lusinoff's experiments were extremely successful in the fact that it was able to create a foundation for the postmodern shellfish industry, we do have hatcheries scattered throughout the country. Those hatcheries are producing individuals which have been able to more or less at least reestablish a stock of sellable oysters. However, there are many major inherent bottlenecks to hatchery production. Food production, microalgae production being obviously the major one. But obviously, there's also a huge concern with being able to predictably spawn and condition oysters. If you cannot get your oysters to produce gametes predictably, you are either A, capped at the amount of production, capped at the amount of season you're able to have, based on just natural gametogenesis of the oysters, which for all the investment that goes into a hatchery, the personnel, the electricity, the infrastructure is simply a waste. But if you're able to control this gametogenesis, if you're able to manipulate it at least enough to extend your season, it can greatly increase the amount of larvae you're able to produce, and therefore the amount of seed you're able to produce, and therefore the amount of profit. The biggest issue that I foresee happening with the industry as is that we frankly have huge deficits in localized hatchery production. Many states altogether don't have a robust hatchery within that state. Be them Rhode Island or Connecticut, and most of them are depending on established large-scale hatcheries up north, up in Maine and in Massachusetts. And if we really do have too few hatcheries, then we really do have too few genetics entering and exiting these farms as a whole, at least guided genetics that are being selectively raised. And if we do that, we do risk the obvious issue of the potato famine, which having an increasingly genetically homologous stock is in the same way begging for a disease outbreak in the same way as having a completely naive wild stock is. Well, as we do with everything, when it comes to being able to control and manipulate the gametogenesis and synchronize the spawning of oysters, it pays a lot to be able to observe what they do in the wild. Thus, this section will be discussing wild spawning behaviors of Crassostrea virginica. Now, in the previous slide, I really wish I'd put quotation marks around wild oyster spawning behavior, simply because to consider Crassostrea virginica as a single species behaving in a single way truly denies the sheer impressive multiplicity of the species. It's been able to expand up and down this coast, all the way down to the Gulf of Mexico, in some cases other countries altogether. But it really does, and it occupies the heads of river systems. So the ability for these critters to withstand a huge range of salinities, temperatures, be able to change their dietary consumptions based on the microbial flux that's going on. There's a huge variance in oyster populations. An oyster collected from the Damariscata in Maine is going to be almost an alien as far as some of its intimate behavior than an oyster that you pluck out of Florida. So here's another quote from Lusinoff that really goes into this diversity. The American oyster is adapted to live in waters with considerable variations in salinity and temperature. Its optimum salinity range is roughly from 10 to 28 parts per thousand. The oyster can survive in the open ocean for some time, but usually it does not reproduce or grow well there. It can also survive periods of spring floods or heavy rains when the salinity of the water is abnormally reduced. The temperature range that oysters can tolerate also varies greatly. For example, in Long Island Sound, the winter water temperature over the oyster beds may drop below 32. While during the warm part of the summer, it rises to about 73 in the deep water and as high as 78 in the shallows. Oysters in the inshore areas of the Chesapeake Bay withstand seasonal temperature variations from freezing in the winter to 90 degrees in the summer. In the Gulf of Mexico, the annual range of temperatures over the oyster beds is from 50 to 90. There's a huge range that these critters are able to withstand. And because of that, the various areas that they're able to colonize means that oyster populations in those various areas behave very differently. So with that in mind, let's make a few points about wild Crassostrea virginica broodstock. Firstly, there's a huge natural range upwards towards the northern parts of Canada down to the Gulf of Mexico. They've been introduced to the West Coast and Hawaii and other places with variable success in establishing themselves. In some places, they're considered invasive. Successful populations will eventually form reefs. You'll have scattered individuals, and then eventually, over time, you'll have re-recruitment occurring again and again and again and again and again through mass spawns, where these petty villagers will prefer to settle in areas with lots of aragonite and calcite. Big piles of oyster shells are good for that, and a mature oyster reef will go through various levels of dead shell, living shell. That shell will die, be re-recruited, and after a matter of time, will be able to preserve itself through recruitment. Because of this huge range, there's a huge degree of mitochondrial DNA variance. And what's meant by that is essentially the oysters themselves, even though they are the same species, they are so distinct in their populations that metabolically they do operate in a distinct way. So the mitochondria, the powerhouses of their cells, are different from one another. So these populations of oysters, even though they are the same species, do behave and have their own genetic distinctions, even to the point where you can derive competent strains, which are unique from the whole. So we'll begin by confronting fundamental issues with classical understanding when it comes to shellfish spawning. So back in the day, in the 1920s, pre-Victor Lusinoff, the understanding amongst the scientific community was that every specific shellfish population, regardless of where it was from or its strain, or as long as it was the same species, had what was, quote, a critical, quote unquote, spawning temperature. That once the water temperature hit this level, oysters will spawn, and that's just the end of that. And that was established by this fellow named Nelson. And of course, we can see here another follow-up study from Victor Lusinoff 30 years later, and that's obviously been made false, where you have various different oysters of various different genetic compositions being exposed to the same temperature, and they definitely have variable amounts of gonad development. So the idea of this static critical temperature is clearly false. Clearly the real answer is one that's a little bit more meticulous and contextualized. perhaps there is a critical spawning temperature, but it's the critical spawning temperature of that oyster population in that particular area, as defined by its evolution as that population has adapted itself to those waters. It is not as clear cut as Crassostrea virginica will spawn at 20 degrees Celsius. This kind of static thinking, I think, is one of the most detrimental aspects to being able to uh, truly understand what's going on with these oysters and manipulate their spawning. You have to be able to be loose a little bit. You have to be looser than the oysters and being able to contextualize what exactly is cueing them. So because we have such a large range for oyster spawning, it's clear that these different populations that have learned to colonize different areas have different reproductive strategies. The same, what the water conditions which are going to be ideal for pediveliger larvae are not going to have the same time scale in Long Island Sound as it's going to be in the Gulf of Mexico. So we see here a couple of general differences between loosely categorized populations of oysters. The first two being temperate versus tropical. We have the oysters that occupy these more warm waters down south in Florida and Georgia and in the Gulf of Mexico, and these are exposed to warm waters year-round, as opposed to the ones up here in Connecticut, Massachusetts, Rhode Island, upwards to Maine, where there really is a seasonal shift in temperature. So when we compare these different populations, we really do see how, particularly, temperate populations, because we're having this seasonal flux in temperature, the seasonal flux in temperature is affecting the stratification of the water column. The stratification of the water column is affecting how much nutrients are being dosed to the photolithic layer, and that is dictating the amount of algae blooms that are happening. The amount of algae blooms that are happening are dictating the successful recruitment of the oysters. So we see here that it's really up to the geological and physical characteristics of the water body that really is going to dictate when the proper algae bloom is going to take place, and that's going to dictate when these oysters have the most reproductive success. And then over the course of years and many seasons, your population is going to adapt a strategy which has their larvae ready when that phytoplankton blooms. So if you're in Long Island Sound, your oyster population is probably going to look for temperature-based cues to spawn. It's going to look for warming waters to associate with the overturn that's going to occur towards the mouth of the rivers and have this big bloom. Now, if you're not going to have that big temperature shift, let's say that you're in the Gulf of Mexico at the head of a river system over there, there probably won't be a huge temperature shift seasonally, but there very well might be a huge salinity shift with the ebb and flow of the river water. So with other oysters, we see in the tropical regions that it's less so temperature-based cues and more so rains and reductions and rises of salinity, which can induce spawning and prepare gametogenesis in these oysters. So based on where our oyster population is and how evolutionarily it has been designed to get its larvae ready for peak phytoplankton, it may determine which strategy will be most efficient as far as being able to convince or trick those oysters that conditions are right for gametogenesis or spawning. So, if they're tropical oysters, we'll probably maybe pick more of a halo shock approach where we shift the salinity rapidly. If they're temperate oysters, much more likely thermoshock. A shift in temperature is going to be the strategy that we adopt. So when it comes to a brief understanding of the formation of gametogenesis in the oysters, and when it comes to spawning, at least what you're going to see when it comes to hatchery culture, oysters are protandric hermaphrodites. They will start off life as the majority as males. Now, as they mature, eventually some of the oysters will undergo sex change and become egg-bearing females. Now, there's also the conditions where, in the absence of enough males, females can also revert back to producing sperm. So when it comes to the actual appearances of oyster spawning, we have the male system, which does not utilize the adductor muscle to ejaculate. It simply comes out as kind of a smokestack of sperm. And this will happen continuously until the gametes are spent. So there's no refractory period. Male oysters will just continuously spew. And this can be very helpful in the hatchery because having a male oyster spew and having his sperm in the water can easily help synchronize the spawn of the rest. But this is in contrast to the female system, which utilizes the adductor muscle to create kind of a shotgun blast of eggs, where you can see this is one of the oysters that I spawned in the bottom right. You can see the eggs are relatively large size and can be seen with the naked eye, even though they look a little dust-like. But there's a refractory period that happens between these egg release episodes. So females will dose the amount of eggs that are released at any given amount of time. So we see here already a contrast between the male and female system as far as how they're going to release their gametes. And this is always also going to be correlated as far as what environmental factors are going to facilitate this gametogenesis. And here we see kind of an abridged version of oyster gametogenesis. At least the various stages can be observed from the hatchery manager slash technician's point of view. So we see that the average oyster that you take out pre-conditioning, it's sexually undifferentiated. So it's very difficult to tell even a male from a female because there just isn't any gonadal tissue visible. By the time that we have early development, we have the follicles in the gonad section beginning to expand and beginning to be able to differentiate into egg producing and sperm producing cells. But it's not until the much later stages of conditioning where you have the gametes beginning to mature in the gonaducts, and you're able to tell sperm from egg, at least once the individual has been shucked. Um, uh, it really, and afterwards, post spawning, you'll see that the gonaducts will empty. Once spawn has occurred, and the gonad tissue will atrophy. So there is a significant refractory period in between spawns. So the concept of reconditioning oysters is very difficult once they've already spawned, because it will take them a good amount of time to produce the energy again to produce gametes, let alone the environmental cues to actually. So in this section, we'll go a little bit more specific to a facility um, scale decision making. So if you're going to have a hatchery, it's indicative. You have to know what broodstock you want, how much of them to get, and when to get them. So that'll be this section. And here, another quote from Lusinoff. Genetics should be one of our tools. A hatchery does not need to keep hundreds of bushels of spawners because even a large one can obtain sufficient spawn from a comparatively small number of clams or oysters. These few mollusks, however, must be selected according to their characteristics, which, as we already mentioned, may include rapid growth, good meat, good shape of shells, and perhaps a certain flavor. So we see here, you know, you 10 to 20 million eggs per female is a conservative number on the last slide. A handful of individuals can produce far more villagers than even the largest facility can house, all the way to seed size. However, to have those individuals that are raised in the hatchery be of the desired genetic characteristics is paramount. And to select broodstock to be broodstock means that you expect that these are the ideal product, not only as far as being acceptable to the consumer, but having the genetic wherewithal to have their offspring be resistant against novel diseases, be tolerant to a wide range of environmental conditions, and to grow quick, have a thick shell, and good tasting meat. And thus it is the task of the hatchery manager to choose their broodstock carefully. Ideally, you're going to want to determine the minimum amount of broodstock that will be needed, simply because these are large animals. They consume an incredible amount of food, even on flow through, and even when supplemented with actual algae, they'll just consume, consume, consume. Um, no real hatchery facility can produce enough microalgae to feed market-sized oysters indefinitely. So it's imperative that a small number of broodstock be selective with the right genetics to produce desirable offspring, but at the same time, you have enough of these broodstocks that you have enough of a genetic diversity to prevent having repeated genes again and again that could make your stock more resistant to one particular problem over another. So 2 to 3 years age is a good rough measure. You're going to want to have some younger ones for males, obviously. You know, the smaller size is best for feed conservation, but it's really those 3-year-olds that have the much deeper shell that guarantee you at least a few females in the mix. And it's imperative that you have a good amount of females because those eggs are going to be one of your bottlenecks. So there are new methods which are being invented. Conventionally, most oyster hatcheries gather enough so that they can at least assure that they will have enough males and females to conduct a successful spawn. New methods are being investigated, such as MRI, muscle relaxants, and also analysis of the hemolymph protein and peptide content to be able to determine sex of the broodstock without killing the animal, and to be able to do that and to be able to ensure a proper ratio so that you're not feeding anyone you're not necessarily going to use for their genetics. And then lastly, local genetics should seriously be taken into consideration because even though it is extremely desirable to have the fastest growing oyster or to have oysters that look like a very marketable version of the competitors, it's imperative that there is not increased genetic homology between all the various farms in the various states. Our greatest asset is the fact that these oysters are inherently diverse and that they have been able to adapt to all these different water bodies. So by respecting the local genetics of each individual water body, we can A, produce oysters most efficiently in that particular water body, and B, we can ensure that we have a vast genetic arsenal to recollect and redeploy should there be another major calamity that wipes out the majority of the wild population again. Here's an example of some acknowledged selectively bred oyster strains, particularly for being able to resist particular environmental conditions and specific diseases, specifically dermo and MSX. So we see here a huge strain, a spread that we have here. We have Maine, Rhode Island, Long Island, New Jersey, Virginia. We see here that after these calamitous events of the wild oyster populations in the 20th century, all these survivors had been recollected. Their genes had been they've been selectively bred so that their offspring were extremely resistant to the perils which had collapsed the majority of the wild population. So in many ways, this list is very inspiring for the ability for oyster farmers to be able to bounce back and be able to revitalize the oyster stocks with these new disease-resistant strains. Though at the same time, it begs the question: if if all the wild oysters have been wiped out by MSX, and now there's a certain population of hatchery-raised oysters that's resistant to this, it would stand to reason that most of the farmers would cultivate these specific strains so that they could have a crop. And if they're relying more and more on these specific strains being produced by a dwindling handful of hatcheries, they are becoming increasingly reliant on a narrowing set of genetics. And this is a trend in and of itself, which may have some blowback in the coming centuries if there is something new that has not been considered in the disease resistant in the selective breeding of these current strains that they could be damaged by. So we see here that these currently established strains are an incredible asset and testament to where the oyster industry has come from, but also kind of a warning sign as to the genetic homogenization that could very well occur because everyone's so dependent on these particular strains now. And so we go over the potential dangers of over relying on these selectively raised stocks. The increased genetic bottleneck that comes from propagating too few strains again and again and again and again and again. And then also the ability for localized genetics that they're so important to retain just so that we can maintain this genetic arsenal should there be a need to develop strains outside of these already derived ones. Obviously, there is a need to reconcile these ambiguously valuable native genes with production standards, and this is the role of the hatchery, to create the best production oyster of the best taste from the genes available in their local water body. This is the best way to reconcile those needs so that those genes are available for the next national selective breeding imperative, while at the same time still having marketable oysters that can be produced. And also increases with the uniqueness of individual brand names. When we go into Lusanoff's thought on the matter, a hatchery may often maintain several stocks of spawners to produce different types of seed mollusks, especially adapted to the needs of the shellfish farmers in different geographical areas populated by physiologically different races of mollusks. This is an extremely important consideration because, as we have learned, some of the races that do well in their native environment grow relatively poorly when transplanted to other waters. Secondly, again by proper selection, the hatchery would develop fast-growing races of bivalves. The same may be said in relation to fatness of meats. Furthermore, special races of commercial mollusks can be developed that may be planted and grown in areas of low salinities or low temperatures where at present their existence is impossible. So we see here another complete consideration altogether with the hatchery and the selective breeding effort, which is the ability to be able to expand oyster farming to areas outside of the oyster's indigenous range. So oysters are not oceanic animals. They are meant to reproduce and grow at the mouths of river systems. Even in salt ponds, their recruitment is relatively poor, even though they can grow there. So if we're going to have races of shellfish that are grown in salt ponds where they're not naturally reoccurring, it stands to reason that one of the other mechanisms that we're going to be keeping in mind is developing broodstock and selective strains that can adapt to newer and newer environments and fulfill the needs of individual farmers in their individual water bodies. And here we'll start to get into the actual practice when it comes to conditioning individual broodstocks in the hatchery and the various processes that can be taken to begin, delay, or accelerate gametogenesis. So when it comes to conditioning oyster broodstock, there's a number of different temperature regimes that must be simulated. So uh, let's go back to the wild oysters. So let's say the Long Island Sound example, where the spawning queue is predominantly driven by seasonal shifts in temperature. So these oysters are sitting here throughout the summer, and they're growing, and they're growing, and they're consuming, and they're growing physically as far as becoming bigger. And it's towards the fall that they begin to stop growing as aggressively. And as the water temperatures begin to cool down, they begin to increase their glycogen reserves and their fat reserves. And it's at this moment that we see the fall period is really when the resources start to get stockpiled within the oyster, and this begins to intensify up until some key winter storage period where the oyster more or less slows its metabolism down and just begins to live off those reserves as the food supply shifts towards unsustainable levels. So the oyster has hopefully consumed all of these resources in the fall and stored them throughout the winter. So as when the spring period begins, a gradual warming is occurring, and at that point, The oyster is utilizing these glycogen reserves. However, they are not using them for somatic growth. They are not using them to become bigger. They are using them for gametogenesis. So at this point, the reserves built up in the fall and you sustain them through the winter are then being used to mature and develop gametes. And then once there is a final conditioning temperature, as this temperature increases throughout the spring, you have increased and accelerated gametogenesis and gonad maturation until there is a key temperature that is reached, at least to start a positive feedback loop of communal spawning. So we see here that wild progression is something that has to be simulated in a convincing way to induce the right cues in the oyster. And there has to be enough time at these specific temperatures to allow the oyster to go through these various stages of cooling and resource storage and then resource reapplication. But at the same time, these conditions still can be accelerated in the hatchery conditions so that this stage of conditioning, as opposed to taking us several months, can take a series of several weeks. So we see here the stages that are attained through the hatchery, where we are taking oysters in, hopefully around the fall and the winter when they have already acquired their key resources. So we're not investing in growing microalgae to regrow all those resources. Hopefully that we are taking oysters during that cool temperature when they already have those nutritional reserves. And then what we're essentially doing in the hatchery is accelerating the warming period and accelerating that spring warming to induce gametogenesis. And once a spawn occurs in the hatchery and in the wild, there is a large refractory period where gametogenesis will likely not restart until the following fall and winter and spring. So with that condition in mind, reconditioning and using the same broodstock from multiple spawns is not widely practiced. And for the majority, it is storing and keeping oysters in the fall/winter condition as cool as possible for as long as possible into the season, so that you have this greatest range. And it's really when selecting these key temperatures and the increments of warming that it becomes crucial to really understand the genetics of the broodstock that you're working with, because each one of these populations is keyed into its own evolutionary advantage of when it thinks that the next grand phytoplankton bloom is going to be in the time of its progeny's metamorphosis. So really at this point, it becomes very important when you're dealing with temperate oysters to really understand the winter, spring, summer progression timing that they're used to, especially so that you can know exactly at which temperatures they will likely be dormant, at which temperatures their gametogenesis will start to get accelerated, and whether or not you run the risk of a premature spawn at a lower temperature, or how severe your thermoshock will have to be for the actual spawning induction. And for tropical oysters, it's been found that for many, especially in the Gulf of Mexico, where you have seasonally warmer temperatures year-round, that they will have just mature gametes year-round, and that their gametogenesis is probably more dictated directly by feed consumption than by temperature cues as well. So while we are reinvigorating the oyster, these winter reserves could be a greater or lesser role depending on the population of oysters that we're dealing with. And as a result of that, it could be more or less entirely crucial to feed broodstock oysters. In some populations, maybe not so much if they're dependent on those winter reserves. In some populations, like the Gulf of Mexico oysters, perhaps it could be extremely important to feed high levels of polyunsaturated fatty acids to ensure that the oysters have these lipid reserves to translate into their progeny to make them as competent as possible. So we look at other extraneous factors that could really affect conditioning. You've taken these broodstock out, you've cleaned them, you've put them in their conditioning tanks, and you're trying to start this temperature progression to induce accelerated gametogenesis. What else can really go happen? What can be wrong? What can go wrong? So one of the nice things about oysters is that they are really highly resistant to ammonia toxicity, both because of the inherent nature of their respiratory physiology, but also because of the fact that they actively denitrify. That goes down. They actually host denitrifying bacteria within their mantle tissue, which allows them to facilitate the conversion of ammonia directly into nitrite and nitrate on site. So they're rather resilient to acute ammonia toxicity, though they will succumb at really high levels. They can spawn in a variety of pHs. It's been demonstrated that they can spawn all the way from 6 to 10, and this is obviously a potential result of their evolutionary context at the mouth of river systems, where there's probably a good amount of pH fluctuation depending on the system. Pollutants have had some indirect, some indeterminate effects on broodstock spawning, but PCBs have been demonstrated to reduce gamete levels in oysters being conditioned. There's also the risk of potential harmful algal blooms. Carenia brevia in Florida, for instance, poses a major issue on flow-through systems in particular. This algae can produce toxins which can hurt oyster broodstock, damage them, weaken them, and reduce gametogenesis, if not cause mortality. And then, of course, there's the constant threat of potential diseases, which could be inversely affecting gametogenesis in your broodstock. Some of these being a little bit more nefarious and subtle than others, such as disseminated neoplasia. It's essentially cancer for oysters. So in summary of the conditioning section, essentially you have to know your broodstock, you have to know the natural context of which their gametogenesis occurs, and then you have to do what you can to recreate that regime in as short a time as possible that will produce reliable gametogenesis. And then we reviewed a variety of different technologies which are also being used to potentially analyze the stages of gametogenesis and the gender of oysters without the need of sacrificing and chucking animals. So once the hatchery has conditioned broodstock that are displaying sufficient signs of gametogenesis, and they have a good gonad somatic index, there has to be a strategy for induction of spawn. Whatever conditioning practices are adopted have to be at the sub-spawn level for if a spawn occurs without hatchery personnel being there, the broodstock will basically just filter out all of the eggs and the trochophores, and all of the effort will have been wasted. So to do this properly, spawning will have to occur when the hatchery personnel are there and can sort the gametes and fertilize them extraneously. And to do that, they want to have as many different gametes going off as possible because the fertilization window is very short. So to do this, they will want to have a synchronized spawn, and to have a synchronized spawn, it is essential to know and be able to deploy a reliable induction spawning mechanism. So the first spawning mechanism is not even a spawning mechanism. It's a forcible collection of the gametes via lethal stripping. So this is killing the animals in order to extract their gametes and basically then fertilize the gametes extraneously. This is commonly used in Crassostrea gyges in France, and also in the production of triploids, where the gametes have to be prepared and controlled in a very particular way. It doesn't matter that fertilization just occurs. So for applications where a high degree of control over fertilization is required, this approach has its benefits. However, for everything else, production-wise, it has several drawbacks. It doesn't allow for reconditioning or attainment of the broodstock. So if you like the result that you get, well, it doesn't matter unless you hold on to the progeny. That broodstock that took 3 years to make is gone because you killed it. Um, which, yeah, again, you spend all these time investing in these animals, and if you have gametes and offspring that you really like, you really don't get more than one use out of these animals. And goodness forbid they are not ready with their gametes, and you kill them prematurely. There's no chance of reconditioning them because you killed them. So perhaps the most classical method, and the method I've used in the past, is thermoshock to induce spawning of oysters. So essentially, this takes advantage of the oyster's natural spawning cycle, at least temperate oysters, which is reliant on gradual increase of temperature, and it shocks and exploits that cycle via shock. So we'll take oysters from 20 degrees Celsius and acutely expose them to water at 30 plus degrees Celsius. So this will hopefully acutely induce spawning within the oysters, though sometimes this is immediate, but however, sometimes it requires many temperature cycles where a half hour, an hour goes by, the temperature must be brought down progressively to 20 degrees, and then another spike, and then if that doesn't work, brought down again. So it can be a 6 minute or 6 hour process, depending on the broodstock and the time of depending on various factors. Uh, so this method is attractive because it only requires temperature control, but it can be extremely tedious if no spawn is induced. So this is hardly a 100% reliable and hardly a 100% synchronous method. So it is fine in certain production settings. However, when it comes to the level of control that will probably be necessary in the future, it does have its limitations. Another method for spawn induction that's been used in the past is halo shock, which is the use similar approach as thermoshock, except you're exploiting a natural cycle of salinity that particularly Gulf of Mexico oysters may be more exposed to and more susceptible to. So this involves dropping the salinity down even as far as low as 5 to 10 parts per thousand, and spiking it again. With towards full strength seawater in the hopes of inducing a spawn. So this was probably more useful again among tropical populations that are less responsive to thermoshock, as producing multiple salinity water is easily more inconvenient than changing the temperature. Once the spawn is induced, so other methods that can be utilized, one that's a little bit more of an accessory method, is once a spawn has been induced in the past, a little bit of sperm will be taken from the spawn bath and frozen. And this frozen sperm will be released into the water to hopefully help induce the spawn of future broodstock. So many times, frozen sperm is used as an accessory technique to thermoshock or halo shock to help increase the reliability of those methods. Something that has 100% or maximum synchrony, or at least the possibility of it, is the possibility of chemical stimulus. So, via a specific technique, serotonin can actually be injected into the gonad of the oyster, and that will induce a spawn. Even if the gonads are not ripe, the oyster will exhibit spawning behavior. So this is potentially a mechanism for 100% reliable spawning induction in oysters. However, obviously it has its own limitations for increased labor, but 100% synchrony could make it highly desirable. Uh, Another method that strays from the everyday production standard but enters towards long-term longevity of perhaps the industry's better interest is prospects of cryopreservation. So there has been success in preserving oyster sperm over long periods of time. So this could really be integral to being able to preserve genetics that may be not favored for, or maybe rare, or maybe less expressed during a given time, but may be incredibly valuable should there be some novel calamity that wipes out the majority of the established oysters, and another hunt for genetics must undergo to find new selective strains that can exist in whatever environment or condition has suddenly occurred. So the idea that you can keep these gametes stored and categorized just like any other high-value crop or seed, or bovine or sheep gametes are stored long period to preserve those industries. So it'd be in the best interest of the oyster industry to probably seek out similar organization. So I understand that in many ways this talk is coming from all directions, but the bottom line is that because of the new nature of the U.S. oyster industry, being that you need a single set oyster that's relatively uniform in size, and knowing the fact that past oyster industries have been completely destroyed by lack of genetic control and malleability of the population, because of these things, hatcheries have to be expanded in number, locality, and scale. Um, we need to be making more oysters for the industry once things recover after this COVID-19 mess. And there's going to need to be the ability to at least have a rough understanding of what genetics are being employed by each hatchery, where those genetics are native to, and then the ability to categorize those gametes based on the traits that might be desirable. Should there be another novel calamity that would require new selective strains that perhaps have characteristics that the current production species, the current production individuals don't have collectively. So the key to having these hatcheries work is being able to condition and spawn broodstock year-round. For production purposes, you invest so much in having these hatchery. You invest in your personnel. You invest in your infrastructure. You want to be able to be producing oysters year-round. You might not necessarily want to for certain reasons, because they won't be able to be deployed out in the grounds, but you want to have the ability to at least to have the ability to selectively breed, and to have the ability to get an early or late start, early start in your season, or make a good season last longer, especially if something erroneous happened early on. For selective breeding efforts in the academic and scientific and government laboratories, it's paramount to be able to breed year-round so that you can truly accelerate the rate at which you're able to develop new strains. And this could perhaps be really essential if we're trying to keep the industry's jobs going if some new disease or the like were to pop up. And again, knowing the intricacies of the localized populations is key. To know what you have, and to be able to produce it, and then to be able to organize the traits of what you have is really the hidden contextual goal of the hatchery, to provide not only the raw numbers of oysters for the production, and not only the quality for the raw bar market, but to ensure that there's at least some knowledge, some understanding of the genetic flow in and out of the oyster population, so that we really can potentially produce something new should the oyster strains that we rely on today be decimated. And with that, I would like to end this talk and end with a picture of an oyster spawn. I'd like to thank you all very much for listening to the talk. Here is a picture of female oyster surrounded by eggs. So happy spawning, everyone, and may you all have many oysters in the future.